We've got people from southern Gibraltar, Kansas, Rio de Janeiro, Tucson. Welcome, everybody. Thank you for celebrating World Oceans Day. It's awesome to get this turnout. And I just got the go-ahead that we are streaming on Facebook Live now, too. So we're going to move forward. I'm going to find my introductions here for our panelists. And first of all, thank you guys for being here today. I'm sure that all of you are in high demand as resources and spokespeople and researchers of our ocean. So it's a treat to be able to connect, which we actually all have connected at Biosphere 2 before. I think the last time I saw you guys was a year or two ago now during a marine ecology workshop for the ocean behind me, particularly looking at ways that we can use this mesocosm at Biosphere 2 to assist in some of your global research in thinking about ways to promote, increase, and study resilient coral reef ecosystems. So this is a treat. I'm going to start with Dr. Stuart Sandin, who is the Oliver Chair in Marine Biodiversity and Conservation at Scripps Institution of Oceanography, UC San Diego. He is a professor in the Marine Biology Research Division, and he serves as director for the Center of Marine Biodiversity and Conservation. Stuart co-developed the 100 Island Challenge, a collaborative effort based at Scripps to describe the variation of coral reefs from across the globe. Welcome, Stuart.
Hey, thank you, Erin. It's great to see everybody and to see all the geographies represented here. I think World Ocean Day is a day for everybody. So you asked to give a background about my entry to the ocean, and my entry to the ocean began on land. I began my career as a terrestrial ecologist, always studying the communities of organisms that live together, but in tropical forests. I worked on both the plants and then the birds that were flying in the forest. That work was in the state of Hawaii in the United States, and it was working with some endangered species. Well, it was during that period of time that I learned that the fate of these birds was really linked to things that were global in nature and local in nature. That work did have me watch species recover in their population sizes, and I watched the species go extinct. So it was pretty dramatic, and that part is part of the reason that I moved to working on the ocean. I saw opportunities in the ocean where we could learn lessons from the land, where we have a longer history of ecology working. So where did I work in the ocean? I started in Hawaii. I moved to the Southern Caribbean. And then when I began my position at Scripps, I began working in the Central Pacific. In each case, I was studying the community ecology, organisms working together. And I realized through that geographic variation that no two coral reefs are the same. That as you go from one coral reef to the next, the systems look different. They each have different flavor and different characteristics. I realized it was that geographic perspective that could help to guide us in understanding what's possible, both what's possible in terms of things going down and possible in terms of our actions to rebuild coral reefs for tomorrow. With that idea about the geographic variation of coral reefs is when I started what Erin just mentioned, the 100 Island Challenge. This is a study like it sounds, to study the coral reefs from 100 different islands, spanning right now 17 countries. And from that, we've been trying to understand the variability. Where's the good, where's the bad? And we see both. We certainly see locations where the coral reefs are going down, where we're watching corals die and the fish is being overexploited. And we see locations, though, where the corals are growing and they continue to persevere. We're even seeing novel adaptations. So while we're seeing this spectrum of variability, the question to me is, how do we use that knowledge? How do we, as good stewards, scientists, and citizens, find ways to solve the problems? I've noted that my colleagues from across the globe have thought a lot about their local situation and how can they use their insights to make sure that the coral reef in their backyard continues to thrive. And you asked me to prepare a statement of the state of the ocean, which I'll give to you now. I'd say that we see fragility in the coral colony, whether it falls underfoot or falls victim to thermal stress. The coral community, however, has distinct strength in its diversity in its ability to adapt ecologically and evolutionarily. It's here that we need to focus our global attention. Happy Earth Day, Oceans Day.
Thank you, Stuart. They're interconnected, the earth and the oceans. Next, I'll introduce Dr. Erin Muller. Erin is a program manager and science director of the Elizabeth Moore International Center for Coral Reef Research and Restoration. She's the manager of MOATS, this is Moat Marine Labs, Coral Health and Disease Research Program, and she was recently awarded the prestigious Presidential Early Career Awards for Scientists and Engineers. Welcome, Erin, and would you share with us your opening remarks?
Yeah, sure. It's a pleasure to be here. Happy World Oceans Day to everybody. So as Erin mentioned, I am a senior scientist at Moat Marine Lab, and I wear a lot of different hats, many of which I really enjoy wearing. So one of them is that I'm the science director of our Elizabeth Moore International Center for Coral Reef Research and Restoration in the Florida Keys. And in that role, I get to oversee all of the coral reef research and coral restoration activities that occur throughout Moat Marine Lab. So we have over 20 different staff members that really focus on coral reef research and restoration in this area, and I get to make sure that that's a coordinated effort. I also have my foundation in coral health and disease, and so a lot of my research from my dissertation and postdoc focused on what makes coral sick and also what keeps certain corals healthy. So as Stuart mentioned, there's a resilience inherently within coral reefs. And what I try to do is try to figure out what are those stressors, maybe they're bacteria, maybe it's related to the environment that are making corals sick, but also what makes corals that survive tick and what makes them so special. And what I really love about my research is now working at Moat, I get to integrate it into my other hat, which is leading our coral restoration efforts. So I help oversee the research in understanding resilience within our populations that are in Florida and to figure out ways to novelly integrate them into our coral restoration plan. So coral restoration has been occurring throughout Florida for the last decade or so, and Moat has been a big leader in this area. We propagate tens of thousands of corals and outplant them back out onto the reef to try to jumpstart population recovery. And part of that process is trying to understand how we can do that the best way possible by creating novel methods to increase restoration success just through the outplanting process, but also trying to figure out how to ensure that the populations we put out there are not only going to survive today, but survive for decades to come. So I work primarily now in the Florida Reef Tract region, and unfortunately that area has suffered probably one of the greatest declines throughout the world. We've lost over 90% of our coral cover over the last 50 years. But the good news is that there are groups like Moat, several of us working in coordination throughout the Florida Reef Tract to try to figure out the best way to bring these reefs back by coupling, policy to try to incorporate climate change policy to try to reduce the impacts of global climate change on our reefs, while simultaneously trying to figure out the best way to bring those reefs back to life throughout the restoration process. So although the reefs, sometimes the state of the reef can be a little depressing to hear about, it's great to be able to try to come up with novel solutions to bring the reefs back so that my children and my children's children have those beautiful reefs to experience like I did when I was younger.
Thank you, Erin. And last but not least, we have Raquel Peixoto, who is an associate professor from the Federal University of Rio de Janeiro and a research associate from the Rio de Janeiro Marine Aquarium, Aqua Rio, in Brazil. She also coordinates the Beneficial Microorganisms of Marine Organisms, BMMO network, which seeks to unify and facilitate studies on the manipulation of the microbiome associated with marine organisms. Welcome, Raquel.
Thank you. It's also a great pleasure to be here and celebrate World Oceans Day. Some of my favorite scientists and people are actually working on things that are very related to what I've been doing. Just like Stuart, I also started in land. I started working on, I'm a marine microbiologist. I started working on beneficial microbes to support some terrestrial organisms and ecosystems. So at some point I started to work also with mangroves. That's when I started to go into the ocean a little bit. And so at that time I started working on beneficial microbes that could help us to restore, and restoration projects correlated with mangroves, highly polluted mangroves that we wanted to recover. And at that point, because we knew a lot about beneficial microbes for plants, we could manipulate those mechanisms that we were aware from the literature and actually make it to support these plants to be restored. But at the same time, because we started working on coral reefs, I started to think whether I could translate some of our knowledge and findings with working on terrestrial organisms into corals. But at that point, we realized that we had no knowledge on these mechanisms. I mean, the specific mechanisms that we should manipulate to promote coral growth, health, or growth. Or resilience. And at that time, because we are all, most of the organisms, including us, are meta-organisms. That means that we rely on symbiotic partners living within us. And so as corals, we need these microorganisms to provide us with vitamins and nutrients and protect us against pathogens. And at that point, I started to think that I could use them. I could use these microbes. And so we started all this work trying to find out which mechanisms, which groups we could or should manipulate. And at some point, we start working in the lab, and now we have like the proof of concept that we can, it's possible to manipulate these microbes and promote coral health, increase their resistance against bleaching and against pathogens and other impacts. And right now we are also trying to understand more about how this process ongoing in corals and how we can improve it to make them grow faster, to make them grow healthier, so that maybe Erin can use some of these knowledge to make sure that her corals will survive once she put them out there. And in Brazil specifically, we have historically escaped the last mass bleaching and mortality events until last year. Last year we broke all the records in terms of degree hitting weeks in Brazil, and we recorded the first mass mortality from Leperosporinis, which is one of the four most important reef breeders in our country. So that kind of show us that we are running out of time. Especially including reefs that we thought that were refugee, that were protected so far. And one important thing for us to have in mind is that even if we stop all the emissions right now, and if we create a different way of life, we still have the committed CO2 in our atmosphere. So we need solutions to make sure that corals will stand until we have a final solution that it's reduced CO2. And my state of union is that coral reefs are extremely threatened, and there is a quite narrow window for us to save them, or at least some of them. At this point, it's now or never. And I mean, we can make it, but we're running out of time.
Thank you guys so much. And it's, just as you introduce yourself, it just feels incredible to have the group of you three here where some of your perspectives can blend together to, try and communicate solutions for reef resiliency, which is the title of our session here. And Stuart, I wanted to start with you. You've done some extensive research in cataloging some of the coral reefs around the world. And as you mentioned in your opening remarks, you said that you're seeing areas where reefs are doing really poor and on their way out, and areas where reefs are growing and in some cases thriving. What kind of patterns do you see in reefs that are thriving versus reefs that are in decline? Perhaps you could describe some of the threats and patterns, whether it be structure and function, or just little things that you've taken away throughout the 100 Island project, in terms of how these reefs are differing, and especially which ones are doing well versus which ones are doing poorly.
Yeah, that's a big question, and one that can take a career to get through. The major threats, indeed, that are facing coral reefs can be grouped into two main categories: the global and the local. When we talk about global threats, those are things having to do with temperature, atmospheric processes, sea level temperature increases, ocean acidification. Local impacts are those that are affecting that have a more narrow spatial range. Things like fishing practices, pollution, and trampling. Those types of activities each interact to influence whether a reef is going to do well or not. The last panel focused very much on those global impacts, and these are ones that have to be addressed for the livelihood of not just our nature, but our humanity. The local impacts are ones that there's a lot more heterogeneity. From site to site, things are different in terms of how we manage the coral reef that's in our backyard. Do we have sewage treatment? Are we respectful about where people put things in terms of coastal development, sedimentation, and in particular, how we exploit those resources? When I say that I've seen things go poorly and things go well, the poorly is ones are reefs that certainly have been affected by some of the global stressors. Those are some of the most obvious. The death of the Great Barrier Reef because of a very large warm water event is obvious. You could see it from a helicopter. But those hot water events also work at smaller scales and are more nuanced. The places where we see things going well are places where there's good stewardship of the local environment, where the fishing practices are somewhat in balance, where we are not doing these direct impacts. The take-home story, I mean, we're in the midst of addressing this, and one of the best things I've been able to learn from the 100 Island Challenge has been to just hear the perspectives from colleagues across the globe. One thing I'll tell you is it doesn't matter what location we go to, from the worst to the best in terms of what you think in terms of health, everybody has a story of a reef that's surprising, that's thriving. And they say it to me almost sometimes like it's a secret, or that they're seeing something wrong. I think there's a lot to this that's happening. I think that one of the reasons we're seeing it is because there are resilient corals out there. Adaptation is happening, and I know that the average rate of adaptation isn't fast, but there are potential to make corals work today, and you can see that. But the second is also our perception bias, is our ability to watch things change. It's really easy underwater to see dramatic loss. Think about watching a forest. It's easy to watch a forest get clear cut and then to lament its loss. It's a lot harder to watch it grow. And we have some challenges in watching things grow. Our local partners who sit there day in, day out are the ones that watch them grow. And sometimes I think those stories aren't amplified. So take home, I think that we certainly have to address both the global and the local impacts. The places right now where we see that probably the shining lights are the ones where the local impacts are well managed.
Thank you. Yeah, it's a really interesting notion that there might be some perception blindness going on, where what we see is driven by various narratives that are out there, and narratives within the coral reef community have been, surprisingly, Erin mentioned depressing in a lot of ways, because we're talking about a lot of loss, and as we'll get into in a second, things like diseases, global and localized threats. So I think it's important to remember that we do tend to seem to focus on what we're losing, as opposed to, really making that extra effort to view some of our oceans and coral reef ecosystems objectively sometimes for the growth that we see as well. And Erin, I want to move over to you. You work on a reef tract that's experiencing a lot of localized and regional disease. Could you describe what's going on with the Florida reef tract, and maybe start to touch on how that's leading you guys towards thinking about solutions for your particular localized events?
Yeah, sure. So Florida, the Florida reef tract has really been shaped by disease events that have occurred over the last kind of 50 years until today. And so a lot of the initial loss that we saw throughout the Florida reef tract occurred in the 70s and 80s, associated with one disease outbreak called the white man disease that affected a lot of the branching species, the elkhorn and the staghorn corals. And that really resulted in its first kind of precipitous decline. And since then, there's been a lot of activity and recording of subsequent disease occurrences that have kind of either maintained an impact on the population and really prevented a lot of recovery from occurring as well. And to kind of make matters worse, right now we're in the midst of an even worse outbreak than what was recorded in the 70s and 80s. It's called the Stony coral tissue loss disease. It began in 2014 off the coast of Miami-Dade County, and has since spread throughout the entire Florida reef tract, kind of precipitating out from Miami-Dade up the northern part of the reef tract all the way to the northern part of Martin County, if you're familiar with the area, and then all the way down into the Florida Keys west of Key West now. So this appears to be a bacterial pathogen. We're still trying to identify what it could be, but it does respond to antibiotics, and so bacteria probably play an important role in this disease. But because of its persistence over the last several years, doesn't kind of wax and wane as some previous disease occurrences have had. And because it affects at least 20 different species of stony corals, mostly the boulder and the brain corals that are really the foundation of the Florida reef tract, it has been termed the worst disease outbreak that has ever been recorded. And unfortunately now it's not isolated to Florida. It's been reported in several other locations around the Caribbean, such as Mexico, Belize, Jamaica, the Bahamas, and the Turks and Caicos. And so we are dealing with this large-scale disease outbreak that's kind of impacting the last vestiges of our corals that we have on the Florida reef tract. But in doing so, it's kind of inspired a community of motivation throughout scientists throughout Florida, and we have had this unprecedented response led by the Florida Department of Environmental Protection. We have a consortium of over 60 different entities that work together to try to understand the pathogen, figure out its transmission. We try to understand how to continue to conduct restoration under the context of the disease outbreak, and really try to combat it with knowledge, science, and a lot of focus. And it's probably been the most coordinated effort in the history of coral reef ecology. And so that has really set a precedent for future impacts that could be happening around the world. But you kind of made a lead into how we incorporate that into what we do within restoration. And this really kind of helps to support a lot of Raquel's work in the sense that we know that there are certain corals, not just reefs, but certain corals that live in reefs that are impacted, that are incredibly more resistant to this particular disease outbreak than others. And their microbiome, their bacteria really seem to play an important role. So we're trying to understand that more as to, is it a certain bacteria? Is it a suite of bacteria? And how do we thoughtfully integrate that into our restoration plan so that the corals that we put out there are the ones that are going to survive this outbreak?
Yeah, Raquel, maybe you could kind of grab the reins here and guide us through a number of things where, how do you go about identifying a coral that is more resilient? And some of the work that you do involves extracting that microbiome, looking at what's in there, and then you say, okay, well, how do we share some of these distinctions with, or can we share some of these distinctive microbial properties with other corals? And you kind of coined the term, coral reef probiotics in a way. In our last session, we talked about this heavy mixing in the ocean too, which, to what Erin was just talking about with disease, I'm thinking, well, if you've got diseases starting locally, they're not necessarily going to stay local at the same time. So how can mixing be both a friend but also a foe when you're thinking about microbial probiotics in a well-mixed ocean?
It's kind of difficult to say that, to only rely on these resistant corals to provide us with probiotics. In fact, what we have is that corals are living there in the ocean, and they live happily associated with their microbiomes. But what happens is that once we have any impacts in the environment and anything that changes, suddenly this community might also change. And that's what we call a dysbiotic process. So all the corals live there with their symbiotic partners until something different happens in the environment. It could be an increase in the seawater temperature, or oil spill, or anything like that that can trigger the change in this community, the composition of this community. And so, for you to have an idea, corals live with this, most of shallow coral lives with these associated photosynthetic algae that in some cases provide up to 80% of the organic compounds that they need. And once, I will give the example of the increasing temperature of the seawater, once the temperature increases and also light, these algae gets overloaded and starts to produce also some toxic compounds, some reactive oxygen species, and this ends up on the coral expels, or the algae leaves the corals, and it breaks down this symbiotic and very important partnership. But not only the algae, other microorganisms also take part of these relationships, as they provide nutrients to the algae and corals, and they make sure that pathogens won't overgrow them. And so this is a very well-established balance. There is a balance between all these players. What happens when we have any disturbance is that we break this balance, and so we start this dysbiotic process. So some diseases are not caused by only one pathogen, and I believe that this is the case that's going on in Florida with the tissue loss disease, because different partners can take part in this process. What we do is that we try to make sure that these beneficial microbes that are out there before the impacts, we want to make sure that we keep them there. And so we avoid this dysbiotic process to start. And that's because these microorganisms are also sensitive to the changes. So what we need to do is to keep providing them to corals during this period regarding bleaching. And in the case of diseases, we need to understand why this community is so unbalanced and provide those specific players that can fight either the pathogens or the community change. We do have, we identified some resistant colonies in Brazil that we are studying. They do have different microbiomes associated with them, and this is very interesting because eventually we have two colonies, same coral species, they live side by side, their mucus can touch each other, and they harbor different communities associated, and they have different phenotypes. So I truly believe that the microbiome is one of the factors. It's very likely correlated with genotypes or anything that is controlled by the host, otherwise they could just exchange this microbiome as they live so close. But what we are trying to do is to crack this code, is to understand how and why certain corals will acquire these specific members of the microbiome that can provide them with some protection, or they can respond better to these impacts and manipulate everything, like the community, the microbes that we are giving to them, the way corals will respond to this manipulation. And so it's a very complex topic, because we just started, and now it's this field is its infancy. We need to understand everything, which are the best players, the mechanisms, how to manipulate it. But to summarize, we need to make sure that we keep these beneficial microbes there once corals are going through any process of stress or anything like that. So the pathogens won't overgrow and the algae will return, or is still there.
Thank you. Our first question from the audience, from Ramya, she asks, can we focus on genetically modified corals that can thrive in high concentrations of carbon? Would the approach give us any evident response? And maybe more generally, you know, within the past several years, there have been terms like supercorals being tossed around. Can we talk about genetically modified corals in the context of what is a supercoral? Do supercorals actually exist? Or is that kind of wishful thinking in some circumstances where there are positives and negatives inherent with any different type of species out there within a different environment? Erin, why don't we move back to you? You guys have been implementing a really wide variety of reef restoration, things like fragmentation. I'm sure you've considered the genetically modified stuff as well. Could you speak to GMO corals and maybe some of the alternative solutions that are out there as well, whether it be fragmentation or fire lining, disease-prone, you know, rain coral? Talk us through some of those solutions.
Yeah, sure. So one obvious step that we need to consider is how to best incorporate resilience, specifically when you're talking about restoring a reef. But one thing that we know inherently is that genetic diversity is one of the best ways to increase the robustness of a population and a community. And so you don't want to necessarily focus on a certain strain of resilience or even a certain coral that maybe you would quantify as a super coral, because you really want to have a lot of genetic diversity, because you can't predict the way that these corals are going to respond to threats that are going to come and go through time. A lot of my research too is focused on trade-offs. And what we have seen, at least in our population of corals that we use for restoration for the staghorn species, is that there is no supercoral, at least in the studies we've done. Disease resistance is independent from heat tolerance, and ocean acidification tolerance may be another response that could be related or independent from those other traits as well. And so, you know, you want to maintain all of those different genetic strains within your population so that as the environment changes, you have a robust population with high genetic diversity. That being said, we do definitely want to think about screening those corals and understanding them more, because you want to have a variety of tools in your tool belt. Like, you do want to be able to think as a scientist, what are our tools that we could use to increase resilience as we're doing restoration? And then you take that the next step as to what are the risks associated with importing those tools into an actual outplanting plan. And so right now we're in the process of identifying those tools. One of those could be, you know, genetically modifying a coral. Another one could be just selectively breeding corals. So just kind of taking a heat tolerant mom and a disease-resistant dad and making babies out of them to increase genetic diversity through their population, but also increasing resilience. So there's a lot of these tools that we're trying to explore as practitioners, as scientists, and also as community members. And, you know, everyday person, we need to incorporate all of the different stakeholders that value those reefs to make sure that any decisions that scientists take are decisions the community would also support. So it's a complex decision process, and we're really just kind of at the forefront of thinking about, you know, the best plans for moving forward and making sure that everybody is communicating appropriately. You kind of mentioned other solutions for things like disease. And so there's been a lot of great movement in that pathway, especially with the Stony coral tissue loss disease. There's been a lot of communication with managers and scientists to come up with innovative ways to try to stop the disease from progressing. So one of the first ways that that has been tested in the field is through a process called trenching. So you kind of create a fire break in between the diseased portion and the healthy portion. And then you can actually apply this paste that's laced with antibiotics directly to the wound. And that has a very slow release antibiotic so that you don't kind of get this big washout into the environment. And it's focused on the lesion, and it's a really small dose. And it works about 80% of the time when it's done correctly. And so that has been used to save really large corals. These corals are ones that are going to release a lot of eggs and sperm every year so that they can, you know, produce the next generation. And so there's been groups that are focusing those efforts to try to save those big sentinel corals that are going to be hopefully driving population recovery after the outbreak simmers down a little bit. And now we've kind of been moving into trying to figure out a whole colony treatment. And this could be anything from like a probiotic treatment, which some colleagues from the Smithsonian, Val Paul and Blake Ushijima, are spearheading to try to treat corals that are sick with a probiotic, and do so that you're treating the whole colony and not just the diseased lesion. And so there's a lot of these really unique, novel ways that we are incorporating now in our response plan, and some that are showing really great promise.
Thank you. In thinking about solutions for reef resilience, I think it's obvious that we have some great humans like you all that are working towards solutions from our point of view. But Stuart mentioned earlier that he's been seeing some novel adaptations, at least in some reefs. What kinds of adaptations and efforts are you witnessing that don't necessarily have to do with our own intervention, but that might be taking place naturally?
Yeah, that's a good question. And then let me preface with, I've got a big dream that we're going to watch all the world's coral reefs, that we can virtually access them, we can interrogate them. We're trying to do that with imagery to bring the reefs home and to share them across the world. I also want to see inside of corals. I want to see their DNA. I want to see their microbiome. I want to be able to see them across the globe so we can answer that question you bring up there, Erin, about the adaptation. I can give you anecdotal reports of what's being seen. There is repeated evidence that locations where there's been a very strong hot water event, for instance, the corals will die. The Phoenix Islands provide a really good test case. If you don't know your Central Pacific geography, follow the equator in between the continents in the middle of the Pacific Ocean, and you'll find the Phoenix Islands. Little dots, low atolls, and it suffered one of the most intensive hot water events. Well, after the first hot water event, the corals most of them died. Well, we went back a few years later and saw little baby corals starting to grow. And they got bigger, and there was some cause for optimism. Phoenix rising from the ashes. And lo and behold, another hot water event. The Central Pacific's really dynamic oceanographically, so it does get particularly hot at different periods. Well, went back and looked to see what happened after that event, and the mortality was not as bad. The number of corals, the proportion of corals was not as bad. We saw a lot of corals survive through that event. And now this is something that's been repeated in multiple locations. Yeah, thanks. This is a nice graphic. It shows things that can happen. If you track here, you've got, this is from Palmyra Atoll to the east of the Phoenix Islands. But what you'll see is a hot water event in 2015. In this case, a lot of the corals regrew. You'll see that they got their tissue back, that where it's white in the middle, that when it gets brown again, that means it has its tissue back. Some of that's because of resistance, meaning that it suffered the hot water and was able to continue growing. In other cases, there was death and then new corals growing. I want to set this up just as a thought experiment. The world has about 250,000 square miles of coral reef. That's kind of ballpark, whatever gets close to 500,000 square kilometers. It's about the area of France. If you start thinking about the area of France and that being scattered across the entire world's tropics, that's a lot of little experimental petri dishes. Each coral reef is a petri dish for thinking about a potential to find a new genetic combination. So the question that Erin just addressed in terms of genetic modification, trying to target the genes, that's one approach, and it's a human approach to try to put particular functionality in. I think that we have a lot of that functionality adapting across the globe in these petri dishes. A hot water event like that is maybe one of the strongest events that leads to survival of the fittest. The only ones that can survive will survive, and then are going to have an open landscape in which to grow. Yes, the average rate of coral adaptation genetically is a lot slower than the average rate of climatic shift. But we aren't thinking about just the average, we're thinking about the extremes. There are going to be cases where you see new genotypes, new combinations. It may be the bacteria linked with the algae that live inside of the coral with the animal, this unique combination that can survive. I can tell you I've been swimming in lagoons in Micronesia where the water was hotter than I would like my hot tub to be. I don't have a hot tub. The hot tub I'm in. And it felt hotter than that. And there were corals growing there. There's something special that can show up. And that to me is not a unique story. That to me, we're seeing in multiple locations, the corals growing back stronger after a second hot water event. The question still remains, will it be fast enough for us during our generation to be able to see coral communities, vibrant coral communities like you see in this video here, persist for our kids and for our kids to tell their kids about? Or are we going to watch a really dramatic few decades where a lot of these communities will be made up of just little ephemeral corals that pop up here and there? How fast will it take to rebuild these ecological communities? I think it's an open question. Where I see some strength is that these little petri dishes with successful experiments pop up in every country, in every island where we've worked. And I'm curious whether with a little help from local human communities, is there a way that maybe these genotypes can spread? Can it be a complementary approach to our active intervention? Can humans play a part in the puzzle to spread? And this could be from Micronesia, French Polynesia, Kiribati, through to the Indian Ocean and the Caribbean.
Thank you. We have a question from Catherine. She says, and probably inspired by this animation, which I just want to say, this is really impressive. Like, we're looking at a three-dimensional model of a coral reef over the course of 5 or 6 years, the same exact spots. That is really cool. Catherine asks, so coral that is bleached can, quote, come back from the dead, so to speak. So can someone make the distinction between coral bleaching as it results in the coral die-off as to a bleaching event and then a recovery from that bleaching event?
Well, as the person who probably knows the least about the physiology of corals here, I'll do a 2-sentence answer, which is that the corals themselves are symbiotic organisms that have algae that live inside of them. It is possible that when they get hot, they expel the algae, and they can, if the hot water, the stress reduces fast enough, slurp the algae back up and not die, bleach and survive. Very often what we see is not happening. Instead, if you see a coral recover, it may sometimes be a loss of coral tissue and a regrowth of coral tissue over the existing skeleton. Little pockets of the coral can survive. Corals, remember, are colonial, and they're a bunch of little guys sitting next to each other, and they can split and grow. So that's a possibility of regrowth as well. I did have a student who tried to study and look for zombie corals. I still think they might exist, but I still might need to just watch some more movies about zombies to really get there.
That's an invitation to all our participants to work on your kind of classic 50s B movie called Coral Zombies. We have a question from Robert C. Robert says, and Robert Christofferson is on the board of the Biosphere. He said, we had 3 international workshops at Biosphere 2, setting a holistic approach to researching resilience in the Biosphere 2 mesocosm ocean, which is behind me. We also had an important advisory board meeting at the Moat Marine Lab this past year, seeing some dramatic work there. Nod to Erin. What progress is being made in selecting which species might be selected for Biosphere 2 research? And we're all affiliated, and you all have kind of a hand in steering and guiding some of our research. So what's the opportunity here? And what progress has been made in that species selection, even if it's just your own personal suggestion?
Well, we at Moat have been talking quite extensively with Biosphere 2 about providing corals. We actually have corals set aside for transfer to the ocean whenever you guys are ready for them. And so we have, we focus on, for restoration, we focus a lot on the major reef building species and also the ones that provide unique niches like the branching corals. And so we have, I think, 4 different species set aside and being grown and ready to be transferred to B2. And then we also have been into discussions about these extremely unique corals that we have at Moat. We were part of an assisted gene flow project, which was a collaboration with Carmabi, which is a lab in Curacao, the Smithsonian, Hawaii Institute of Marine Biology, Florida Aquarium, and Moat, where we actually used cryopreserved sperm from Florida and Puerto Rico and crossed it with fresh eggs from Curacao to create these novel genotypes as a way to test the methodology for moving fresh genes from other places in the Caribbean into Florida, because the elkhorn coral is basically functionally extinct within the Florida Reef Tract, unfortunately. And so we're trying to find tools and novel ideas to increase the genetic diversity of our population. And we right now have hundreds of these corals that we're looking for homes for. And so we have offered them up to the biosphere to integrate within the ocean as part of that novel plan moving forward.
Yeah, in our workshop a couple years ago, we threw out the term Franken Reef, which I loved and fits into our zombie movie. We may not run with that idea right off the bat, but it's awesome to be able to set up some partnerships with you guys and to potentially gather some coral from around the world to see how these different species might interact with each other for the first times. Let's see, we've got a question from high school biology teacher, Casey White. How would you go about realistically addressing the future of coral reefs with kids? And what can they do to preserve the reefs? Raquel, do you want to take a stab at that?
No, just it's a coincidence because we just had a paper out, Frontiers for Young Minds, specifically talking about coral probiotics and all the problems of coral loss in the world. This is specifically prepared for kids and reviewed by kids as well. And it's quite interesting to see that, first of all, they are way more open to change their way of life than adults, of course, because they are more like flexible and they are super concerned about the world that they will get from us. So in terms of changing the way of life and what kids or all of us can do, it's more or less the same things that we talked in we heard from the previous section on with making small efforts that can be very significant if all of us start to do that, as we can use less our cars, we can bike more, we can walk more, we can reduce our meat consumption, we can stop or reduce the amount of meat that we have. We can move from red meat to white meat, chicken, salad. We can become vegans as well, because we can also work on the plastics as well, because not only of the pollution in the oceans, but also because the way they are produced and the hydrocarbons behind it that can also contribute to reduce CO2 emissions. Education and spread. I mean, kids can have this very powerful communication to reach out to adults, to their parents, to convince them to change small things in a daily basis. I think that this is what kids can do. There are some material and some videos and some material for kids, specifically designed for kids out there, where they can research and learn a little bit about what's going on and what can be done. The work with probiotics, in our case, we used a very fun parallel with M&Ms, which is mutualistic, these microorganisms that we are using, these mutualistic and symbiotic microorganisms to make this very fun. There is a video showing how corals are bleaching and the way we think that bacteria could provide a shield for these corals to resist thermal stress and pathogens. So I think that it's pretty straightforward for kids to take this material and work on these better than us.
And I'll just mention that I'll follow up with Raquel afterwards, and we'll present some of these links that have been offered up, both here and also from the past discussion, references to some of the things we're talking about here. So stay tuned for that. Now, Stuart, I want to turn back to you because in addition to the work that you've done on coral reefs, you also have this great background with fisheries and fish. And there was a paper that came out by a scientist named Jeremy Jackson a while ago that basically stated that ecological extinction caused by overfishing trumps any other threat to our coastal ecosystems. Now, I was wondering if you could talk about the relationship between fish and coral reefs, whether it's a symbiosis, and how threats like overfishing might play a role in some of the challenges that we don't necessarily think about right away.
Yeah, that's a great question, and it really does bring up the thought that coral reefs are not a bunch of isolated organisms, but instead they're integrated communities. Jeremy Jackson's wife, Nancy Knowlton, would push back, and the statement is coral reefs are the rainforest of the sea. She says rainforests are the coral reef of the land, because coral reefs have been there longer. That you can argue on your paleoecology, but there's something really true about the integration across the different groups of organisms that live on a coral reef. To focus solely on the coral animal, to think that it's just this passive agent that's responding to the climate and nothing else matters is absurd. It's naive, and I don't think we would think that about any organism or any ecosystem, to focus on one organism. What do fish do on a coral reef? Fish do a lot. They eat, and they eat particular things, and the way that they eat is what's important. Probably the most important element in a coral reef that's linked to the role of fishes is what they eat off the bottom. If you ever go to a reef and you watch it for a while, one of the things you'll see happen quite a bit is things in the water column eating, that they're eating plankton. That's the little stuff that's floating through the air. But then you'll see a lot of the fishes going to the bottom and poking their head down. Well, they're eating algae. So corals are animals that have algae inside of them, and then there's other types of algae that live next door to them. Well, if nothing eats algae, it grows. And if algae grows too much, it becomes competitive. Your lawn, if you don't, I'm not going to get any sort of analogy. Everybody's seen outdoor things. If you mow one half of a lawn, you don't mow another, one's going to be taller. It's pretty straightforward. Well, there's a lot of importance to the issue of mowing the lawn in a coral reef. Algae can contribute to some negative effects on corals, really locally, in some important ways. Like Raquel is talking about, the microbiota that lives on a coral is linked to how much algae is nearby. Whether they're putting direct chemicals, sugar, whatever, it changes that microbiology. So in short, you can just think that these lawnmowers are helping to contribute to what does the bottom of the reef look like, and making perhaps a better environment for a coral to grow. There are other things that happen on coral reefs that go further up. There's a lot of statement. I know Jeremy Jackson will talk oftentimes about the roles of top predators. Top predators are important because they affect the behavior of smaller predators, and they affect the behavior of these herbivores. There's an intricate food web. Sharks are part of a food web. Big snappers are part of a food web, as are these planktivores. There's a ton of interconnections on a reef, and the studies right now, it's a very active area of research. So if there are any people out there looking for what's the next thing to study, thinking about the interaction between fishes and the corals is an open area. Whether it's the role of this herbivory, or eating the algae on the reef, through to the other organisms eating, and where their excrement goes. There's an ongoing conversation about whether there's moving nutrients, because a fish eats here and then pees inside of a reef. Is that good nitrogen or bad nitrogen? Corals need it. In short, corals, the coral reef ecosystem evolved through time with fish around, big fish, with predators around, with big herbivores, or these algae-eating fishes. The competitive dynamics of a coral versus an algae, or even corals versus corals, are influenced by the structure of that food web around. When we start pulling out entire chunks of the coral reef community, it's not surprising that the coral community itself is going to go through some shifts. It's a new environment now, and there'll be different winners and losers. Jeremy emphasizes that importance of these consumers, and I think he's right. We'll never get a reef back if it has no fish. But I also think we won't get a reef back if it's too hot. For us to solve these problems with reefs, for us to really be looking towards resilience, we have to be attacking both fronts. We have to be making sure that the climate is flattened. We flatten the curve. And we also need to be making sure that we keep the community structure, the fishes, the bacteria, all the other players, looking somewhat like where these organisms evolved.
Thank you, Stuart. I've got some birds in the foreground here, and I think they were communicating with the birds in your square, talking back and forth to each other. So it's not just human species that are communicating on World Ocean Day, it's bird species as well. Erin, I want to flip back to you in light of what Stuart's talking about, where there's really much more of an interconnectedness going on the Florida Reef Tract. Do you guys think about the history of fishing in Florida and how that might have affected the current situation? Do you incorporate natural histories into your research with disease and other issues that you're dealing with?
Yeah, absolutely. We have to think about it every day. And there's there's a lot of good stories about the Florida Reef Tract too. So, there's no doubt that the fish community has changed, that the fish are much smaller, they're less abundant, but there has been good management put into place over the last decade or two that has shown some stabilization and recovery of some of these important reef fish. And so that's kind of a really nice example of good management practices, good buy-in with the community, and nice recovery. But one of the challenges we also have is that even with all of that great management in place, we still have too much algae on the reef, and we have a lot of sediment too. And that can add like another negative feedback into the potential for recovery, because the substrate that is there, that's supposed to be nice and clean for corals to grow on, is not healthy. It's covered in sediments, turf algae that can trap the sediments, that can actually abrade the coral and in disease. And there's a lot of macroalgae that is still on the reef. And so we definitely need to think about how do we clean up that substrate so that there is vast open areas for these coral babies to settle on, to grow and survive. Because we don't see that happening, and that has led to us having to grow them in the lab in order for us to outplant them at a bigger size, because as little babies, they're not able to combat those issues that wouldn't be there if the environment was healthier, if the water was cleaner, and if all of those fishing communities were still in place. And so we have a lot of different, sorry, my daughter's in the background watching her iPad, laughing at something. But we have to, think about it from all these different issues and really engage our management to help us attack it on all fronts.
Thank you. I want to flip to a slide, this one, for Raquel. This looks like some kind of a baby coral, or at least a fragment of a coral. And this is a photo from Raquel's laboratory, where I'm just guessing she might be adding some sort of probiotic solution to a coral fragment. I want to talk about, what kind of thought have you put into how you would deploy a probiotic kind of technology like this? What would that kind of intervention look like as it plays out in a more regional coral reef ecosystem?
So that's a very good question. We've been working a lot on that. We've been developing some strategies that we think that could be efficient to deploy these probiotics in the field. We've worked with bioremediation of oil before. I started working on, and we still work a little bit on that. So we had some kind of ideas that can be adapted from these applications to coral reefs, but specifically focused on reefs and restoration. We've been developing some strategies where we use rotifers to carry our alive probiotics. So we just drop these in big tanks, and corals actively eat these rotifers that will carry these probiotics into the system. There are other things that we are developing as well, such as bacterial pills that we prepare, and these pills will slowly release bacteria into the water, and so we can just spread this on the reef. We can do this by air, or we can use robots that can go and spread these bacterial pills. These bacterial pills will adsorb to coral mucus or sediment, and will keep releasing this bacteria over time. So we don't need to do this all the time. You can deploy it once every month or something like that, until you have the thermal event gone, or the disease outbreak. So these are some of the things that we've been thinking. There are other crazy ideas that we are developing with some engineers, but it's doable. And the most important thing, and I think that this is a key aspect of this technology, of this strategy, is that we don't need to keep applying probiotics in the reef all the time. We need to do this just before we have a thermal event to happen, and we know when they are about to happen because we have NOAA alerts every year. And so we need to go there just before, inoculate it, and wait for a month or two, or work on this for these 2 critical months. So it's not something that we need to be stable forever in the reef, but just before and during this thermal event.
Thank you, Raquel. And we are closing in on our hour. In fact, we're probably spilling over just a little bit. But I do want to end thinking about, what do coral reefs look like in the future? And we may get some grim responses and we may not, but maybe we'll start with Stuart. What is a coral reef? What do our coral reefs look like 100 years from now?
Yeah, I'll answer that 2 ways. If I'm just asked to give percentage breakdown, my go-to is I expect 1/3 of them to be transformed and unrecognizable, scorched earth. I expect 1/3 of them to be struggling along and kind of adapting. I expect 1/3 of them to look somewhat like coral reefs of today. The reason is adaptation. I think that every reef has that potential, and I think that's one of the important points. We are seeing different thermal stresses, and some of the models will say, hey, this area is going to get hotter and this area is going to get less hot. I don't think we know where adaptation is going to happen. I don't think we know where the local human environment, those local stressors, will be well managed. And I can tell you that the people I know living in each island never want to be on a side where science no longer goes saying, hey, we have a chance to do something better with the environment that's off our shores. So I've got a lot of faith in the human spirit that's happening across the globe in terms of fighting to find a solution, from the pipettes like we're seeing in the image here, through to the communities who are trying to find whatever guerrilla warfare to keep coral reefs around. So my pessimism is third. I hope for better.
Erin, do you want to chime in? What does the Florida Reef Tract look like in 100 years?
Yeah, I have a lot of faith that all of the effort that we're putting into the research and the practice today is going to pay off for 100 years from now. And that's why, that's why I have the job that I do. And that's why everybody, that studies coral reefs and studies coral restoration, I think that's what we're all banking on. That's what we're dedicating our life's work to, is to figuring out ways to make sure that in 100 years from now, we still have coral reefs that we would deem healthy and hopefully thriving. So I have that hope that the Florida Reef Tract looks way better 100 years from now than it does today, that it may look different than what it did in the past, but that it's one that's, when you dive in under the water, is breathtaking. And I hope that in 100 years from now, they look back and say, wow, I'm really glad that they fought for this.
And Raquel, any of your own words for what's happening, at least within the regions that you work in, or more broadly, what do coral reefs look like 100 years from now?
Yeah, I fully agree with my colleagues. I think I'm optimistic, and I think that I see how the community has been working hard and together to change the prognostic and to make sure that we still have our corals. I think that we just had this huge change of pattern due to COVID-19, so we changed our social behavior a lot in the last few months, and I do think that maybe this is it could teach us a lesson on how hard it is to live. It's not to be able to use our home, our planet, and I think that this could help us now to have more support, to get more support from the community and the society and stakeholders and governments, because now we know how the apocalypse looks like, and so we can have like a glimpse about how important it is for us to not only preserve corals but all other ecosystems that are threatened in the world, because this is all connected, not only to pandemics but also all other problems and services that these ecosystems provide. So I'm hopeful that we learn something, and that all the efforts that all the scientists and governments are putting on restoration and solutions will pay off.
Well, you guys, we've been getting a lot of participants leaving messages in the chat, thanking you all for doing this today. People from STEM fields, whether it be teachers or students, citizens. So I think your presence here today with us is greatly appreciated, and I want to thank you for doing this. And this will be a great resource for everybody to have. We're going to post both of these sessions up on YouTube for people to revisit or visit for the first time, and be sure to share these talks with your friends, to all the participants out there. It's just been rich in knowledge and solutions and outlook going into the future. So happy World Ocean Day to all of you guys, and thanks again for joining us, and look to Biosphere 2 to continue to host these types of events, but also things like podcasts and hopefully some in-person events moving forward as well. So the outlook looks bright, especially with the work that our panelists are working on and the work that's being done here at Biosphere 2. Thank you guys so much, and go Oceans.
Thank
you.
you. Thank you, guys.
Happy World Ocean Day.
Ocean Day. Bye. Good to see you all.